How chemical changes to DNA help scientists estimate how the body is aging beyond calendar years. Why age is more than a number. When people talk about age, they usually mean chronological age, the number of years since birth. However, two individuals with the same chronological age can have very different health conditions. One may have strong cardiovascular health, good mobility, and low disease risk, while another may show signs of accelerated aging. This difference led scientists to ask an important question. Can we measure how the body is actually aging rather than just counting years? The concept of biological age refers to how old the body appears based on physiological condition rather than time. It reflects the cumulative effects of genetics, lifestyle, environment, and disease exposure. Researchers have developed different ways to estimate biological age, including markers such as inflammation levels, metabolic function, and cellular health. Among these approaches, epigenetic clocks have emerged as one of the most precise tools. What is epigenetics? Epigenetics studies changes in gene activity that do not alter the DNA sequence itself. Instead of changing the genetic code, epigenetic mechanisms control how genes are turned on or off. One of the most studied epigenetic modifications is DNA methylation, where small chemical groups attach to specific regions of DNA. These patterns of methylation change over time and can reflect the aging process. How epigenetic clocks work? Epigenetic clocks analyze DNA methylation patterns at specific sites across the genome. Scientists have identified particular regions where methylation levels correlate strongly with age. By measuring these patterns, researchers can estimate biological age with surprising accuracy. If the epigenetic age is higher than chronological age, it may indicate accelerated aging. If it is lower, it may suggest slower aging. Why DNA methylation changes with time? As cells divide and respond to environmental factors, methylation patterns gradually shift. Exposure to stress, diet, toxins, and physical activity can influence these changes. Over time, the accumulation of these modifications creates a measurable signature. This signature acts like a molecular record of how the body has aged under different conditions. Applications in health and medicine. Epigenetic clocks are being used in research to study aging-related diseases such as cardiovascular conditions, cancer, and neurodegenerative disorders. By comparing biological age with chronological age, scientists can identify individuals at higher risk for certain diseases. These measurements may also help evaluate how lifestyle changes or medical treatments influence aging processes. Implications for preventive health. Understanding biological age could improve preventive health care. Instead of relying only on age-based guidelines, doctors may eventually tailor recommendations based on how a person's body is aging. For example, someone with accelerated biological aging might benefit from earlier screening or targeted interventions. This approach can make healthcare more personalized. Limitations and ongoing research. Despite their potential, epigenetic clocks are still being refined. Different models may produce slightly different estimates, and researchers are working to improve accuracy across diverse populations. Scientists are also studying how reversible these epigenetic changes might be and whether interventions can slow or even partially reverse biological aging. The future of aging measurement. As research progresses, epigenetic clocks may become more widely used in clinical settings. Advances in technology could make testing faster and more accessible. Combined with other biomarkers, these tools may provide a more complete picture of health and aging. This could shift how society understands age, moving from a simple number to a more detailed biological profile. Final thoughts. Epigenetic clock measurements represent an important step in understanding how the body ages. By analyzing chemical changes to DNA, scientists can estimate biological age with increasing precision. This approach offers new insights into health, disease risk, and the effects of lifestyle.